well, that 10% that's left, we don't know that it's a problem. It might work just fine. That's dumb. If 90% of your protein is gone, it's obviously a problem. So one of the things that I do all the time is go through people's genomics reports. Why? Because there is buried gold in there. Usually there's something that's been completely missed that makes a major, major, major difference in the treatment options and hopefully the treatment plan for that person's cancer. So why do I say this and why am I talking about this now? Again, I do this all the time, and some things come in clusters. What I've been finding recently is a large number of oncologists, if they look at the genomics reports at all, they will highlight the things that are flagged for them. And these reports aren't set up to flag the most useful pieces. They're flagged for the most well-known pieces. And that can set you up to chase something that is utterly useless for you. Let's not have that happen to you. Your genomics report is gold. Let's make sure that it's interpreted well for you. You get the full value out of it. Hello, and welcome to Elevating Cancer Treatment, where we explain the science and debunk myths to help you navigate your health journey. My background is a little different. Beyond educating about cancer, I'm actually designing new drugs that are defining the future of oncology. This direct, hands-on experience offers me a very different perspective of how these cancer treatments work on the body, interact with the cancer cells, and cause side effects. And these are insights that I'm excited to share with you. If that sounds interesting, make sure to like this video, subscribe to the channel, and hit that notification bell so you never miss an update. And please share it if you find it useful. I'm Dr. Chaplin. An important reminder, I'm a PhD, not an MD. The information in this video is education, and it's not medical advice. Every cancer is unique, and no general information applies to everyone. Please remember that. Always consult with your healthcare provider for guidance on your specific situation. And two quick things. First, as a thank you for being here, I've created a free resource, 10 Things to Elevate Your Chemo Journey, which you can download from the link below. And second, by signing up, you'll also get updates on that innovative cancer treatment I'm working on. I'm confident it represents a significant advancement in immunotherapy. So please take a moment, download your free guide, and join us in shaping the future of cancer treatment. So these genomics reports, in many cases, they have an astounding amount of information. And the report that you read and that your oncologist looks at is not something that's compiled by a human being. It's compiled by a piece of software. And how does the software put it together? It's getting all of the sequence data, hundreds of thousands, millions of reads of all sorts of different genes from your DNA, from the tumor DNA. And it's trying to pull out what's changed, what's not changed, and what you should pay attention to. How does it tell you and your oncologist what to pay attention to? And where does that sometimes go wrong? Here's what it does. It looks for alterations that have been shown in the literature for your particular type of cancer to have an impact on cancer. That specific mutation in that specific cancer, and it will then highlight it even more if there is a particular drug or clinical trial relevant to that. And that sounds great. That's a great first approach. But in my experience, that often leaves 90% of the value buried down below, because where the real value is in things that aren't immediately obvious. So let's talk about some client stories. I have a client, well, I have multiple clients who have melanoma. One of these clients has 6 different mutations in DNA repair pathways. They have 3 of them in the BRCA genes, 2 in BRCA2, 1 in BRCA1. Those are classically seen in breast cancer, not in melanoma. So they're buried down in the bottom because it's not melanoma. Those genes are breast cancer genes, not melanoma genes. So they're down in the bottom. And they're down in the bottom for another reason. They've also got another 3 mutations in DNA repair pathways that are more common in lung cancer. So again, down in the bottom. The other part is these are mutations that aren't normally seen, and so they are listed as what are called variants of unknown significance, VUS. Stuff buried in the bottom is usually in this category of VUS. We don't have 30 research papers on this specific mutation, so we can't say whether it's actually a problem or not. That's what VUS means, and that is a dumb category. I'm sorry, that is a dumb category. In some cases it makes sense. If there's a particular amino acid, proteins are made of a string of amino acids. They're the building blocks. There's 20 different kinds, and the order in which they're put in determines what protein you get, how it folds up, what it does. So if you change one amino acid, yeah, it might make an impact, it might not make an impact. Sometimes it can be hard to tell. For that, it makes sense. In this client's case, most of those changes were changes from an amino acid to a stop codon or a frame shift, because the translation from DNA to protein is you take 3 letters of DNA and that turns into 1 letter of protein. So if you lose one here, everything shifts. It's like taking 1 letter out of a book and shifting everything down, but keeping all the spaces the same. It turns into garbage, turns into gobbledygook. The stop codon is a very similar thing. Its DNA then says, don't put another amino acid, just stop here. So these kinds of errors shut down the protein early. And in this client's case, you've got scenarios where this is a variant of unknown significance. We don't know if this is going to cause a problem for the protein. This is a 2,000 amino acid long protein, and 150 amino acids in, it just stops. Let's cut 90% of your car off with a giant chainsaw and throw it away. And then we're going to say, well, that 10% that's left, we don't know that it's a problem. It might work just fine. That's dumb. If 90% of your protein is gone, it's obviously a problem. This client has 6 errors that very obviously hit DNA repair pathways. They are not normal DNA repair pathways for melanoma. They're common to other kinds of cancer, but we have drugs for them. These are now off-label drugs, and it can be difficult to get reimbursement or insurance coverage for them, but we know that these drugs will work. Any one of those 6 mutations would make this client a candidate for the class of drugs called PARP inhibitors. If you have damaged DNA repair pathways and you block a different DNA repair pathway, you can actually make it so hard on the tumor cell that it just suicides out. This is a really good approach, and it works beautifully with these drugs in breast cancer, in prostate cancer, in lung cancer, when you have these mutations. But these mutations, he only needed 1 of them. He has 6 of them, but they're not listed because they're not melanoma and they're not exactly the mutations that we normally see. That's gold. Another client also has melanoma. Melanoma tends to have a lot of mutations. With this client, again, same sort of thing. There was a mutation in a gene called IDH1. That's normally seen in glioma. But again, we've got an excellent drug for that. It does really, really, really well. It works in glioma. It works in any cell that has that mutation. But because glioma is not melanoma, it was not up at the top. It was buried in the bottom, which most oncologists don't read through. If you've got a 50-page report, are you going to read through anything more than the first page? It's kind of like your Google search. Who goes past the first page of your Google search hits? Nobody. So this is why having really good interpretation of your genomics reports makes a huge difference. And here's another piece. And this is, honestly, it's a hidden secret that I'm going to tell you about that shouldn't be hidden. This should make total sense. When you get back one of these genomics reports, there is a percentage listed for each one of these genes. And it's got a little acronym next to it. It can be very opaque. A lot of oncologists don't even know what it means. It's the VAF, variant affected fraction. This is the amount of cells in the tumor that have this mutation or this change. Why is this important? That's not one of the things that the software that generates the reports filters on. So you may have something in that headline section that is exactly the mutation that they were expecting for exactly your type of cancer, and it may have a drug that is stellar for it. But if that variant affected fraction number is 5%, sometimes I've seen it as low as 0.2%. Let's say it's 0.2%. That drug is fantastic. It will kill those tumor cells like that. 100%, 100%. But you're only killing off 0.2% of the tumor because only 0.2% of the tumor has that mutation. And almost no one looks at the variant affected fraction, the VAF percentage. If you're going to take a drug to treat your cancer, and you've got a genomics report that you're basing these choices off of, you want the variant affected fraction number to be as high as possible. At least 20%. I would say at least 50%. You really want to go for high percentages. You want the things that are common to most of your cancers so that that drug can do a large amount of damage to the cancer. You don't want to go after 5% of your cancer or 10% of your cancer. You want to go after 80%. And again, that number is largely ignored. It's ignored by the software, it's ignored by most oncologists, it's even ignored by a lot of genomic counselors. It is critically important for your interpretation. Please go through everything in the bottom section. Go through and make sure that those variants of unknown significance aren't hidden gold for you. And when you find something, make sure that it's a target and a drug that's expressed through most of your cancer so that it's actually going to be of benefit to you. These are things that everyone should be looking for in their genomics report, and it's things that every oncologist should be flagging in the genomics reports. So again, I'm fully aware that my experience and the experience of my clients is biased, because they're not going to come to me if they've had a great experience and a successful experience with their oncologist. And I find a tremendous number of opportunities have been missed, and a tremendous number of very small value leads being chased like they're the most important thing. And this to me is really sad, because it means that many people are not getting the quality of treatment that they really could get or deserve. So that's part of why we're putting together, and we're getting close to done, a completely separate package that's essentially a canned course in being able to interpret genomics reports and to know which ones to look for, because the quality varies radically across genomics reports. Some are great for some things, some for others, and some are just awful across the board. They look slick, but they're not really worthwhile. So everything from what kind of sample you need, whether you can do blood, whether you can do tumor, what type of genomics test to do, what the different protein biomarkers are, what the different numbers mean, how to interpret that. This is something that we can do for you one-to-one as a client, but it's also something that we can educate you for. And we'll be putting a link to that product in this video as soon as we get it finalized. Thank you. Beyond these videos, if you need more personalized guidance or a deeper dive into specific treatments to have your treatment be as effective as possible, I offer one-on-one sessions and medical advocacy. You can find information on our website, which is linked down below. Again, if you found this video informative, please give it a thumbs up, click the notification bell, and subscribe to our channel for more science-based cancer insights.